Okay, so my genetics class started talking about this technology called CRISPR on the first day of school. So it must be somewhat important. It uses part of CRISPR sequence of DNA to genetically engineer the base pair of a gene. So you're probably wondering what that actually means. To give an example, if you test your unborn child to see if they have the high chance of contracting a genetic disease, you can edit their base pair so they'll be healthy when they're born. Now, to completely honest, this is neither a good nor a bad thing, but right now, there are more positives that outweigh the negatives. When put in the situation to choose whether to fix their baby or let them lead an extremely suffering-filled life, any parent would give anything they could do to relieve the difficultness of a child's life. It's just what parents do. There are about 6,000 known genetic disorders, but many more just waiting to be discovered. For example, Huntington's is a well-known neurodegenerative genetic disorder that affects muscle coordination and leads to cognitive decline and dementia. Although usually occurring between the ages of 35 and 44 years old, this can be identified as early as even before the baby is known to be a baby. Genome editing is a very controversial, versatile subject that is talked about and the subject of many conversations. People wonder if we should be able to edit human embryo. Should we be able to wipe out an entire species? Because that is what we have the power to do right at our fingertips. This technology is moving faster than people care to recognize and care to pay attention to. Once this technology was brought out, there was no bringing it back. There's only moving forward from here on out. But people need to understand the technology before using it. People need to find the ins and outs of what it is capable of doing, what and where it's going. This technology first damages the DNA. It's a double-strand break through the double helix, then the cellular repair process kicks in, and we're able to actually condense the repair process to make the edit we want as opposed to the natural edit that would usually occur. It's a two-part system. There's the Cas9 and the guide RNA, and together it's combined for CRISPR. It only is a 20-letter target system, which is easy to design and easy to control. It's cheap to buy in comparison to all the other projects that are going on, and it's very efficient. Instead of taking the weeks that it would normally take, it takes days. This cuts down time and saves time, which saves money. And on top of that, it's very powerful. As I mentioned, we're unable to know every detail about CRISPR, but that's what our goal should be. Our goal should be to understand where it's going, where we can take it. But for that, we need money. We need people that are willing to search it for their entire lives and dedicate time. But with the time and the money needed in laboratories, we have a life-changing technology. Life-changing. That word means something to the people it changes lives to and the people around everywhere. The guide RNA and Cas9, they go about the genome until they reach a place where the guide RNA matches and inserts itself between the two strands of the double helix, ultimately ripping them apart. This triggers the Cas9 protein to cut, and the first responders are called. At first, the DNA is taken and puts them back together, which isn't really efficient because a base may drop out or a base may be added that wasn't necessarily wanted to be added or taken out. So then a second repair pathway takes a homologous piece of the DNA to repair, and it takes over, and it feeds a false piece of DNA so you can put whatever you want in to trick the cell. You can change the letter, or you can take a letter out, or you can put new DNA in. And this catalyzes scientific advancements. This leads to our future. It leads to what we're going to be made of eventually. We need to be able to figure out what it's capable of. And to do that, we need to know the power of CRISPR. And the power of CRISPR is the future.